Hello, my name is Marco Benavides, and I'll be presenting on predicting RNA-binding protein interactions using hybrid neural networks. So RNA-binding proteins are critical regulators in biological systems. They influence RNA expression, translation, and also gene silencing. But predicting RNA-protein interactions could be and can be challenging. Now, the model I base my work on is the IDPS. It uses convolutional neural networks with fully connected layers and a bidirectional short-term memory to predict if RNA binds to specific RBPs. The paper uses 31 datasets, with some datasets overlapping. And the IDPS predicts whether an RNA will bind to a protein or not. This is based on the sequence and the secondary structure. Now, my proposed model is a 3D RBP. It introduces several advancements. It integrates a CNN with a transformer, incorporating positional embeddings to capture sequence dependencies. Some key improvements include using not just RNA primary and secondary structures, but also protein features such as amino acid sequences, secondary structures, and tertiary atomic coordinates. Now, the main advantages of the 3D RBP are that it addresses imbalances in binding and non-binding data across all proteins. It also includes both RNA and protein information, providing a more complete feature set. The transformer also allows for better long-distance interaction, modeling positional embeddings. And unlike the IDPS, which uses separate models for protein, the 3D RBP employs a single model for all proteins. It also aims for a higher area under the curve and improved generalization. The dataset that I used include 86,000 RNA protein samples. The RNA structures came from the Vienna RNA, while protein structures were sourced from either the protein data bank or AlphaFold if there was no files from the protein data bank. Initially, datasets were highly imbalanced, with non-binding samples outnumbering the binding samples, but this also extended to protein secondary structures, showing a bias towards coils. However, nucleotides were even, but hairpins were notably overrepresented. The method that I used started also with a baseline CNN model for binary classification, but then I balanced the data to improve the predictions. I then included 3D protein data and later introduced a multi-class approach using a softmax activation layer to predict these 34 classes, which are 17 proteins and their binding, non-binding counterparts. Finally, I added a transformer with multi-head attention and positional embeddings to long-range dependencies. Now for the results, early models like the CNN and balanced CNN showed high accuracy for binary classification, but adding multi-class prediction significantly lowered the accuracy. For the final model represented in blue, achieved an area under the curve of 98.41%, and also the class-specific accuracy revealed variability, with some proteins performing well and others very poorly. Also, the confusion matrix showed a clear diagonal line of correct predictions, but also two off-diagonal trends. These likely represent false non-binding and false binding classifications. Finally, the IDPS served as a benchmark, achieving an area under the curve of 86%, but my model's binary classification also matched this area under the curve, and the multi-class version achieved an area under the curve of 98.41%. For future interactions, we should optimize the transformer by testing hyperparameters like attention heads and layers to improve performance, but I also think it's very important to clean the data, so reducing inconsistencies by selecting reliable datasets and standardizing which experiments to test for the proteins. And finally, we could also incorporate protein surface properties and evolutionary data for better future representation. Thank you.